Hello and welcome back to the Shaky Science Show, or the Spooky Science Show, I should say today. As since Halloween is just around the corner, I thought I'd do a Halloween special. So, I don't think I would really describe myself as a scary person, so I really struggled to think of what to talk about in this video. Should I talk about the scariest proteins, how many bacteria in your body, the effects of alcohol and liver damage? I don't really want to get into that one. But actually, there was one disease that is truly scary, and it's a disease that currently there's no cure, and that is Alzheimer's disease. So, Alzheimer's disease is the most common neurodegenerative disease, and it's predicted that by 2050, 135 million people will be affected by the disease, and with a cost of $30,000 per person for treatment in a year, that could toll up to $4 trillion, a huge amount and a great burden to our society. But whilst these high costs might cause some fear for people, the truly devastating outcome of Alzheimer's disease is the effect on memory, that is, memory loss. And this is because memory makes our lives, enables us to recognise people, make associations, hold conversations, reminisce, have nostalgia, tell funny jokes, and not surprisingly, memory is the key to Pixar movies. My favourite animation films, of course. So I've actually made two previous videos that look at both the biochemical underpinnings of Alzheimer's disease and also memory formation. But in this video, I wanted to really focus on a study that looked at the role of irisin in Alzheimer's disease. And predominantly, I'll be focusing on the results from this Nature publication here. And this paper came out earlier this year and makes a connection between an exercise-induced hormone and memory formation in Alzheimer's disease models. But firstly, a bit of background to Alzheimer's disease. So Alzheimer's disease results in a deterioration of the brain, in particular a region of the brain called the hippocampus. And as I already mentioned, it's the most common neurodegenerative disease, and it's characterised by the presence of these protein aggregates, either amyloid beta plaques, which are found outside the cell, or neurofibrillary tau tangles that are found inside the cell. And so the condition can either be caused through familial and inherited defective genes, or they could happen sporadically. And so it's been reported that there is a link between increased amounts of physical exercise and potentially a decreased rate or incidence rate of getting Alzheimer's disease. So this paper tries to further explore this link and does it by looking at the link between irisin and Alzheimer's disease. So, well, what is irisin? So, with its name derived from the goddess Iris, messenger of the gods, and also the goddess of rainbows, hence I can finally use my rainbow pen, irisin is a myokine. It's a hormone secreted by muscles. And so the signal comes from the muscle and can then travel, is thought in the current model, to the brain, where it can further signal that exercise is being undertaken. So the name comes more from the messenger side than anything to do with rainbows, unfortunately. And irisin isn't just made as irisin, it is derived and cleaved from a precursor that is referred to as fibronectin type 3 domain containing protein 5, or FNDC5. So the idea is that irisin then travels up to the brain and stimulates the expression of brain-derived neurotrophic factor, BDNF, which is involved in learning and memory. And so that's kind of the link between the two so far. But one of the problems with irisin being derived from a precursor means that detecting irisin as opposed to detecting FNDC5 can be quite tricky. And that's important when we want to understand the signalling mechanism involved with irisin signalling. Nonetheless, the team addressed that this was an issue and they tried to be aware of this throughout the study. So what they first did was try to see how do these levels of irisin vary in patients with and without Alzheimer's disease. And what they could see is patients with Alzheimer's disease, and also in mouse models that are meant to mimic Alzheimer's disease, they see lower levels of irisin. In particular, the levels of irisin were lower in the cerebral spinal fluids of Alzheimer's disease patients. And via both genetic and pharmacological approaches, they reduced the levels of irisin in the brains of normal mice, and this appeared to impair their certain types of memory. And then the reverse effect of increasing the levels of irisin boosted their memory and rescued these impairments, which kind of proves the link between irisin and memory defect. And then one of the really neat things that they did, as can be seen in this graph here, is that they gave recombinant irisin in conditions with and without these alpha beta oligos. And what they found was the recombinant irisin actually rescued the impaired neurotransmissions caused by the alpha beta oligos. And so that kind of also shows a link between Alzheimer's disease and irisin levels. Now, whilst it could somewhat be argued that these are more correlations than necessarily causations, one of the interesting parts from the study is the question of, could irisin be used as a biomarker? And also, could exercise be more encouraged as a potential therapeutic to tackle the onset of Alzheimer's disease? The issue with that is obviously dementia is an age-associated condition, and there's also other comorbidities as well, such as arthritis, heart disease, obesity, and so exercise isn't necessarily the most ideal solution. But could we mimic the effects of irisin? And so to be able to mimic the effects of irisin, we kind of really need to understand what is irisin interacting with, what is the receptor that it's binding to in the neurons, if there is a receptor. And so these additional questions need to be answered before we take this any further. But it's pretty cool, it's pretty neat, and shows that a lot of work is being put into Alzheimer's disease. So hopefully at some point, and some point soon, we can have a better cure for Alzheimer's disease. So hopefully this has helped you have a good introduction to where we're at, and thank you for listening.